All right, welcome back to the deep dive. Today, we are taking on a mission that is, well, it's absolutely foundational for anyone heading into clinical medicine.
It really is.
We're doing a deep dive into the principles of infectious disease, pulling everything we can straight from Chapter 6 of Davidson's medicine.
Yeah, think of this as your boot camp. If you're a medical student, you're about to hit the wards, this is the blueprint.
It can feel like a lot, though. The sheer volume of information, all the different agents, the immunology, the drugs.
It's overwhelming, and that's our goal today, to synthesize it. We want to find the anchors, the metaphors that make these essential concepts actually stick.
Exactly. This isn't about reading a textbook out loud. It's about building a kind of cognitive map so you can actually navigate this stuff.
And the map, the big metaphor we're going to use for this whole deep dive, is learning the rules of engagement.
The rules of engagement. I like that.
It's an eternal war, right, between the host, your patient, and the microbe. So we have to identify the players, understand their weapons, learn how our diagnostic toolbox works, and then finally figure out how to deploy our own arsenal strategically.
Okay, let's start right there. With the language of the battlefield. Who are the players, and what do we actually mean when we use a word like infection?
Right. It's a word we throw around a lot.
It is. But in the hospital, it has a very, very specific weight to it.
Yeah.
So for a student, what is that strict clinical definition of infection? How is it different from, say, a microbe that's just along for the ride?
Okay, so in the strict sense, the clinical definition of infection is a process where the microorganism isn't just there.
Not just hanging out.
Exactly. It has to have established itself in the host. It's replicating, and this is the key bit. It successfully induced a host response that leads to pathological change.
The damage. That's the dividing line.
That's the line, the harm, the pathology. That's what separates a disease state from just normal residents.
Okay, so if there's no pathological change, that means the microbe is just, coexisting. And that brings us to colonization. Why is that distinction so important?
Because colonization is, for the most part, a healthy coexistence. It's when a microorganism survives and replicates, but it's doing it on an epithelial surface.
Like on your skin or in your gut.
Exactly, your skin, your mouth, your gut lining, but it's doing it without causing disease. We're colonized by trillions of these things. Bacteria, fungi, viruses, and that's our normal flora. Most of the time, it's actually helping us.
And then there's the third state, which is, It's much more sinister. It's the hidden enemy, latent infection.
Latency is all about dormancy after an invasion. The organism got in, it invaded host cells or tissues, but instead of causing a big acute problem, it goes quiet.
It's waiting.
It's waiting. It's surviving the host's immune surveillance, ready to reactivate if or when the host's defenses drop. Classic examples are tuberculosis or the herpes simplex virus. They're established, but they're not causing active disease yet.
That's a critical distinction.
Yeah.
And we also need to separate how the organism got into the fight in the first place.
Right, which is the difference between an infectious or communicable disease and an endogenous infection.
So communicable is person-to-person spread?
Yep, directly or indirectly. But an endogenous infection is caused by an organism that was already part of your normal colonizing flora. It moved.
It translocated.
Exactly. Think of a classic urinary tract infection. The E. coli causing it was probably living harmlessly in the large bowel, but once it gets into the bladder, a place it's not supposed to be, it becomes a pathogen.
And for those communicable diseases, the text lays out that classic chain of infection. You need a portal of exit, a mode of transmission, and a susceptible host.
If you can break any single link in that chain, you stop the spread. It's the fundamental principle of public health.
Now, before we get to the agents themselves, we should probably touch on the historical standard for proving a microbe caused a disease, Koch's postulates.
Yeah, from the 19th century, they were incredibly rigorous. You had to find the organism in the diseased host, isolate it, grow it in a pure culture, then introduce it to a healthy host and cause the same disease, and then re-isolate it.
But the text says they're now seen as more of a historical standard, not a mandatory checklist. Why is that?
Because the field just got too complex. A lot of really important modern pathogens just don't fit that mold.
Like what?
Well, some organisms, like the one that causes syphilis, Treponema pallidum, are basically uncultivable on artificial lab media. So you fail right there. And then you have all the endogenous infections we just talked about, where the organism is part of the normal flora, which violates Koch's rule that it should only be present in the diseased host.
So we needed better tools.
We needed molecular methods, which we'll definitely get into later.
Okay, so the first key takeaway here is a big one. Not all microbial presence is disease. Colonization is normal, even beneficial, until that organism translocates or overgrows. Infection requires actual pathological change.
That's the core concept.
All right, let's meet the combatants, starting with the smallest. We're calling viruses the cellular pirates. Why that metaphor?
Because they are the ultimate hijackers. They are obligate intracellular parasites. They have absolutely no ability to replicate on their own.
There's just a package of genetic material in a protein coat.
That's it. And they have to subvert the host cell's machinery to make copies of themselves. Their genomes are also incredibly varied. It could be single or double-stranded DNA or RNA.
Yeah, we can't forget that special subclass that gave us one of the biggest health crises in modern history, the retroviruses.
Right. Retroviruses like HIV, they're the real genetic rebels. They carry an enzyme called reverse transcriptase, and what it does is it converts their RNA genome back into DNA inside the host cell.
Which then gets integrated into our own chromosomes.
And that's how you get latent long-term infection. It becomes part of the host.
So, structurally, every virus has that protein coat, the capsid. But it's the presence or absence of a fatty outer layer, the envelope, that seems to dictate so much about how they behave.
Oh, this is a massively important clinical distinction. Enveloped viruses, think influenza, HIV, coronaviruses, they have this outer lipid membrane they steal from the host cell.
And lipids are fragile.
Very fragile. So these viruses are easily destroyed by heat, by drying, by disinfectants. That means they have to be transmitted through close contact. Respiratory droplets, body fluids, they just don't survive well on surfaces.
Which is the opposite of the non-enveloped viruses, the tougher ones.
Exactly. Non-enveloped or naked viruses like norovirus or poliovirus, they don't have that fragile lipid layer. They are much, much hardier. They can survive drying detergents.
Which is why they spread so easily through the fecal-oral route or on contaminated surfaces.
And it's why a non-enveloped virus like norovirus can shut down an entire cruise ship. It just persists in the environment.
If we follow the viral life cycle that's laid out in the book, it really does look like an act of piracy. It all starts with specificity, right?
It starts with adsorption, or attachment. The virus uses proteins on its surface, like the spike protein on SARS-CoV-2, to latch onto very specific receptors on a host cell. That binding determines which cells it can infect.
Then it gets in penetration
Yeah.
and releases its genetic material uncoating. And then our cell's machinery gets put to work making viral parts.
Right, synthesis, followed by assembly of all the new little virus particles.
And then the grand finale, release.
Which can happen in two ways, either by budding, or an enveloped virus pinches off and steals a piece of the host cell membrane on its way out,
Lysis.
or lysis, where a non-enveloped virus just bursts the cell open, which, as you can imagine, causes a lot of inflammation and tissue damage.
Okay, let's move up in size to the autonomous agents,
Mm.
the prokaryotes, or bacteria. They can reproduce on their own, but clinically it seems like everything comes back to their cell wall.
It really does. The defining feature is the peptidoglycan cell wall. It's the target for so many of our best antibiotics. In gram-positive bacteria, like Staph aureus, that layer is really thick. It's like a suit of armor.
But in gram-negative bacteria, like E. coli, it's different.
It's much thinner. And critically, it's surrounded by an outer membrane.
And that outer membrane contains the infamous lipopolysaccharide, or LPS. Let's just unpack why that one molecule has such a massive clinical impact.
LPS is a huge part of why gram-negative sepsis is so dangerous. The lipid component of LPS is what we call endotoxin.
And it's not secreted by the living bacteria.
No, it's part of the structure. It's only released when the bacterium is damaged or dies, often when it's attacked by our immune system, or even by antibiotics. And when it's released, endotoxin unleashes this massive, nonspecific inflammatory cascade.
We're talking vasodilation, fever, clotting problems.
All the hallmarks of septic shock. It's that one structural difference that can determine the severity of the entire systemic response.
Beyond the main chromosome, bacteria also carry these little extra rings of DNA, the plasmids. How important are they in this war?
They're the currency of resistance. Plasmids can carry genes for antibiotic resistance or new virulence factors, and they can be passed between bacteria, sometimes even between different species.
So that's how resistance spreads so quickly.
It's the primary mechanism. This horizontal gene transfer allows them to evolve countermeasures to our drugs at an incredible speed.
Now, identifying these bacteria used to be a pretty slow process in the lab.
Oh, yeah. The classic toolbox. Gram stain to see the shape and wall type, looking at them under a microscope, seeing how they grow on an agar plate. It gave us clues, but a definitive ID could take days of biochemical testing.
But the textbook makes it clear there's been a revolution in identification.
A revolution powered by speed. The biggest game changer is probably MALDI-TOF MS.
Can you break that down?
It's basically protein fingerprinting. You take a bit of the microbe, you zap it with a laser to turn its proteins into ions, and you measure their mass. It compares that mass profile to a huge database and can give you a definitive ID in minutes.
Minutes instead of days.
It's a massive shift. And you couple that with things like 16S RNA gene sequencing for the weird bacteria that won't grow, and whole genome sequencing, and that diagnostic delay is just shrinking and shrinking.
Okay, last group. The more complex eukaryotes. So fungi, protozoa, and helminths. They're structurally more like our own cells, which makes them harder to kill.
It does. For fungi, the main clinical players are yeasts and molds. The key thing to remember is that their cell membranes contain ergosterol, not cholesterol like ours, and their cell walls have 1,3-D-glucan.
And those are our therapeutic targets.
Exactly. If your drug targets ergosterol or glucan A, it should leave human cells alone. It gives us that selective toxicity we need.
And the parasites, protozoa and helminth.
They're often characterized by these really complex multi-stage life cycles that might involve different hosts or vectors, which makes controlling them more of a public health and epidemiology challenge.
All right, time for a critical check-in for you, the listener. The difference between gram-positive and gram-negative is absolutely fundamental. So can you recall the key structural difference in the cell wall? And more importantly, why is that component in the gram-negative outer membrane LPS so medically important? If you can't immediately bring to mind that thin peptidoglycan layer and the role of endotoxin in causing systemic inflammation, just hit rewind and listen to that section again. It's that important.
Definitely.
Okay, so we've identified the players. Now let's shift to the battlefield itself, the host and the weapons they deploy. We're framing the normal microbial flora as our home security system.
Which is a great way to think about it. This system is it's immensely complex and powerful. Our heavily colonized sites, the gut, the mouth, the skin, they host this incredibly diverse ecosystem that really works in our favor.
And the text points to 3 main benefits.
Yeah. Beyond the obvious stuff like making vitamins K and B12, our flora is essential for just priming and maintaining a normal immune system. But its most direct defensive role is something called colonization resistance.
So it actively keeps bad bugs out. How does it do that?
A few ways. It outcompetes them for nutrients and for places to attach on our cells, but it also actively changes the local environment, like lowering the pH in the vagina to make it inhospitable, or even producing its own natural antibiotics, which are called bacteriocins.
They're basically defending their turf.
They are. They're keeping the neighborhood safe from invasive species.
And we have to anchor the difference between these colonized sites and the truly sterile sites. Where should we absolutely not find any microbes?
Right. Submucosal tissues, the bloodstream, the fluid in your joints or around your lungs, these places have to be sterile. If we find bacteria there, it's a sign of pathology. Full stop.
Now, places like the lower airways or the bladder are a bit different.
They're heavily protected. You have the mucociliary escalator in the airways, sphincters in the urinary tract. They might have a very limited transient microbiota, but significant colonization is blocked.
But sometimes that home security system fails. An endogenous infection happens. And it's usually through one of 2 mechanisms, either overgrowth or, more commonly, translocation.
Translocation is the key. It's the physical spread. If a normal skin bug, like Staphylococcus epidermidis, gets a direct route into the bloodstream through an IV line, that's translocation. It's now in a sterile site where there's no competition, and it can cause a serious infection.
And this is especially common in patients who are immunocompromised or have some kind of physical breach.
Exactly.
So given how protective our flora is, the idea of trying to restore it when it's been damaged has become a huge area of medicine. The book mentions probiotics, and then the more dramatic one, fecal microflora transplantation, or FMT.
FMT it's an extreme example, but it's incredibly effective. You're basically taking the entire healthy gut ecosystem from a screened donor and transplanting it into a patient.
And the main proven benefit is for recurrent C. diff infection.
That's its home run indication. For recurrent Clostridioides difficile infection, the success rates are dramatic.
Why does it work so well for C. diff specifically?
Because C. diff infection is the classic example of what happens when colonization resistance fails. Broad-spectrum antibiotics wipe out the good guys in the gut. which allows the resilient C. diff spores to germinate, multiply, and start pumping out their toxins.
So FMT just puts the good guys back.
It immediately and massively restores the competition. It doesn't necessarily kill the C. diff directly. It rebuilds the entire ecosystem, effectively starving it out and restoring that natural protective environment.
Okay, let's switch to the offense. The pathogen's weapons. First, we need to classify the enemies. Primary versus opportunistic pathogens.
This is a classification based entirely on the host state. A primary pathogen, like Mycobacterium tuberculosis or Shigella, can cause disease in pretty much any healthy person it encounters.
It doesn't need an invitation.
Not at all. But an opportunistic pathogen, like Staphylococcus epidermidis or Candida albicans, only causes disease when the host's defenses are compromised.
An opening.
Right. It needs an opening, like an IV catheter, a prosthetic joint, or some form of immunosuppression. That distinction is absolutely critical for how you think at the bedside.
And a pathogen's ability to cause harm, its virulence, comes from its toolkit, its virulence factors.
Mm-hmm.
We've talked about competition, but what about the physical weapons?
The very first weapon is the adhesin. This could be a bacterial pilus or a viral protein, but its job is to allow the microbe to stick to a host cell and resist being washed away. But the most formidable weapon, especially for device-related infections, is the biofilm.
Let's expand on this idea of the microbial fortress.
Yeah.
What exactly is a biofilm?
It's not just a pile of bacteria. It's a complex, living, three-dimensional community of microbes, and they're all encased in this protective slime matrix that they secrete.
What's the slime made of?
It's a mix of polysaccharides, host proteins, even bacterial DNA, and it allows them to stick incredibly tightly to surfaces, especially things like plastic and metal.
So catheters, prosthetic joints, heart valves.
Exactly. And the clinical significance is huge. That matrix physically shields the bacteria inside. It stops immune cells from getting in. And, crucially, it severely limits the penetration of antibiotics.
Which is why these infections are so hard to treat.
It's why they're so notoriously resistant. You often can't cure a biofilm infection with antibiotics alone. You have to physically remove the device, the source, to have any chance of success.
Okay, from physical structures to molecular weapons, the toxins. We already drew the line between endotoxin and exotoxins. Let's just reinforce that.
Right. Endotoxin is the structural LPS from gram-negatives. It's released when the cell dies, and it causes that generalized, chaotic, inflammatory firestorm of sepsis. Exotoxins, on the other hand, are proteins actively secreted by living bacteria, and they're often incredibly specific.
You give us some examples of that precision.
Think about the exotoxins from C. difficile. They specifically target the lining of the large bowel, causing the classic pseudomembranous colitis, or the neurotoxins from the bacteria that cause botulism or tetanus. They have incredibly specific targets in the nervous system. They're like guided missiles, whereas endotoxin is more like a bomb.
And then we have the pathogens that don't fight out in the open, the intracellular pathogens.
These are the masters of hiding. This includes all viruses, obviously, but also bacteria like Salmonella and Mycobacterium tuberculosis. They've evolved to survive and replicate inside our own cells, often inside the very immune cells meant to kill them, like macrophages.
Which protects them from antibodies and a lot of drugs.
Exactly. It's a protective niche. And that's why treating something like TB requires such a long multi-drug regimen. You have to get the drugs inside the cells and kill the hidden bacteria.
And finally, what drives this whole arms race is genetic diversity.
Constantly. Bacteria using their plasmids, viruses with their incredibly high mutation rates, they are constantly swapping genes and evolving. This is how they acquire new virulence factors, or more commonly, antibiotic resistance. It's how new epidemic or pandemic strains like we saw with SARS-CoV-2 can emerge so quickly. Okay, time for another important check-in. Imagine a patient has an infection on a prosthetic heart valve, and the bug is Staphylococcus epidermidis, a common skin organism. Is that microbe acting as a primary or an opportunistic pathogen? And why is the presence of that biofilm structure so critical for how you think about treatment? Just take a moment. The foreign material, the valve, is the opportunity, and the biofilm is the fortress that makes treatment so challenging.
Right. Okay, so once we suspect an infection, the clinic and the lab have to work together. What are the three main goals of the investigation?
It's simple, really. 1, confirm there actually is an infection. 2, identify the specific pathogen. And 3, figure out its antimicrobial susceptibility. You need to know who the enemy is and what bullets will actually work.
And before we find the bug, we can confirm the war is happening by looking at non-specific markers.
These are the signals of generalized inflammation. We look at the white cell count, the C-reactive protein, or CRP. In really sick patients, we might look at lactate, which is a sign of poor tissue perfusion.
The text also mentions procalcitonin, or PCT.
Yeah, PCT is sometimes used to try and differentiate bacterial from non-bacterial inflammation, though its role is still debated a bit. The key thing to remember with all of these is that they are non-specific. A major trauma will also give you a sky-high CRP.
Okay, and the classic symptoms are fever and rigors. I've always wondered, why do you shiver so violently when your body is trying to get hotter?
It's a fascinating mechanism. Rigors, that uncontrollable shivering, happen because your body's central thermostat in the hypothalamus has been reset to a higher temperature
By what?
by circulating pyrogenic cytokines, things like IL-1, IL-6, and TNF, which are released in response to microbial products like endotoxin. So your brain now thinks your normal body temperature is too low.
So it panics and tries to generate heat.
Exactly. It initiates massive muscle contraction shivering to generate heat and drive your core temperature up to that new, higher set point. It's all based on a central misperception.
That makes so much sense. Okay, let's get into how the lab actually finds the culprit.
The fastest answers often come from looking at the sample directly.
Starting with the old school method, microscopy.
Right, just looking for the whole organism. We use bright field microscopy with stains. The most famous is the Gram stain, which separates bacteria based on their cell wall structure. For bugs with waxy walls like TB, we have to use special acid-fast stains.
And sometimes we use dark field.
Yeah, for really thin, delicate organisms like the spirochete that causes syphilis, it makes the organism light up against a dark background.
But sometimes we're not looking for the whole bug, we're just looking for a piece of it, an antigen or a toxin.
And this is the basis for a lot of our rapid tests. Think about the rapid antigen tests for SARS-CoV-2, or tests that look for Legionella antigen in urine, or, crucially, for the actual C. difficile toxin in a stool sample, which is what's causing the disease.
But the real game changer, the DNA fingerprint, has been nucleic acid amplification tests, or NETs. Why are they so powerful?
Because of the power of exponential amplification. A NET, like PCR, uses specific primers to find and make billions of copies of a tiny piece of the pathogen's DNA or RNA. So you can detect incredibly small amounts of the organism.
It's fast.
Super fast and super sensitive. Their biggest use is in virology, where viruses are hard to grow and colonization isn't usually an issue. In bacteriology, they're essential for unculturable bugs, or for testing sterile sites like blood or spinal fluid.
And you can even do a broad-range search.
Right. We can use a broad-range bacterial PCR that targets a gene all bacteria have, the 16S RNA gene. If that's positive in a sterile site, we can then sequence it to figure out exactly what rare or unexpected bug we're dealing with. And we can also use NETs to look for specific resistance genes, like the MECA gene for MRSA.
But despite the speed of NETs, we still need to do culture, the growth test.
Why? Because you need the live bug. Culture is the only way to get a pure isolate of the organism that you can then use for antimicrobial susceptibility testing, or AST. The big downside is, of course, time. A fast bug takes a day, a slow one like TB can take weeks.
The book highlights the process for blood culture, which is so dependent on good clinical technique.
Absolutely. You need a sterile technique, you need the right volume of blood, you need multiple samples. The bottles go into an incubator and are automatically monitored for growth.
And once that growth flag pops up,
The old way was a Gram stain and then days of biochemical tests. The new way, as we said, is MELD-TOF-MS. You can get a definitive ID in minutes.
that's a huge deal for patient care.
It's massive. It means you can de-escalate your antibiotics from broad to targeted so much faster. And taking multiple samples is key to tell the difference between a real, persistent bloodstream infection and just a bit of skin contamination when the needle went in.
Okay, but what if the bug is gone, or it's just impossible to find directly? Then we have to look at the host's memory, which is serology.
This is all about detecting the host's antibody response. The key terms here are titers, which is how we quantify the antibody level, and seroconversion.
Which is the gold standard.
It is. It means you've gone from having no detectable antibody to having a detectable level. Or, more commonly, you demonstrate a fourfold rise in the titer between a sample taken early in the illness, the acute sample, and one taken a few weeks later, the convalescent sample.
And the type of antibody, IgM versus IgG, tells you about the timing.
Right. IgM is the first responder. If you see IgM, it usually means a very acute or recent infection. It tends to fade away after a few weeks or months. IgG is the memory antibody. It comes up later but sticks around for a long time, indicating past exposure or immunity.
The diagram in the book about the window of opportunity is great because it shows why a single IgG result can be tricky.
Yeah, if you test too early, you might miss everything. If you test too late, you might only see stable IgG from an old infection. That's why that fourfold rise in IgG between the two paired samples is so powerful for proving a recent infection.
The main method for this is ELISA.
The enzyme-linked immunosorbent assay, it's a workhorse. It uses enzyme-linked antibodies that create a color change, and you can set it up to detect all sorts of things.
And for really specific confirmation tests.
We use an immunoblot, or Western blot. It first separates all the microbes' proteins by size, and then it checks to see if the patient's antibodies bind to specific protein bands. It's very specific, often used to confirm things like Lyme disease.
And then there's the one we're all familiar with now, lateral flow tests.
The rapid point-of-care strips, they're cheap, they're fast, but you do sacrifice some sensitivity for that convenience.
Okay, here's a check-in on that diagnostic timeline. Imagine you get a positive IgM result for a virus. Does that likely mean it's an acute or an old infection? And why is showing that fourfold rise in IgG between two samples also so important for diagnosis?
Yeah.
The IgM points to an acute infection, and that rising IgG is the proof that the immune system is actively responding to a new challenge right now.
Exactly.
Okay, final step in the toolbox. Antimicrobial susceptibility testing, or AST. We need to know if our weapon will work.
Right. AST tells us if the organism is susceptible, meaning its growth is inhibited by a concentration of the drug we can safely achieve in the patient.
And the key metrics are MIC and MBC.
The minimum inhibitory concentration, or MIC, is the cornerstone. It's the lowest concentration of the drug that stops the bug from growing. The minimum bactericidal concentration, or MBC, is stricter. It's the lowest concentration that actually kills the bug.
But the report I get back doesn't just list the MIC. It tells me if it's susceptible or resistant. That's based on the breakpoint.
The breakpoint is the critical link between the lab and the patient. It's a preset threshold based on clinical outcome data. If the bug's MIC is at or below the breakpoint, it's called susceptible. If the MIC is higher, it's resistant.
And how does the lab figure this out visually?
The classic way is disc diffusion. You put a little paper disc with antibiotic on a plate covered in the bacteria. The bigger the zone of inhibition, the clear circle where nothing grew, the more susceptible the organism is.
Or for a more precise number.
We use diffusion strips, or E-tests. They have a gradient of the antibiotic on them, so you can read the exact MIC value right off the strip where the zone of growth meets it.
All right, we've found the bug, and we know it kills it. Now let's zoom out to the population level. How do infections spread, and how do we stop them?
This is the world of epidemiology and infection control.
We start with classifying patterns. An endemic disease.
That's a disease that's just always present at some level in a particular geographic area.
And those patterns are not static.
Not at all. The book points out that things like climate change, allowing the skeeters to move to new areas, or human migration patterns bringing resistant bacteria across borders, these things are constantly shifting the map of endemic disease.
And then they're the ones that catch us by surprise. Emerging and re-emerging diseases.
An emerging disease is a new one, like SARS-CoV-2 jumping from bats to humans. A re-emerging disease is an old one we thought we had under control that's now coming back, often because of things like falling vaccination rates, like we're seeing with measles.
The source where the agent lives and replicates is its reservoir.
And that can be a human, an animal, which gives us zoonoses, or just the environment. And the concept of the asymptomatic human carrier is so important for diseases like TB or MRSA, where people can spread the bug without being sick themselves.
Okay, let's talk about the actual transmission routes.
Yeah.
The book lists the big ones, but I want to focus on the vehicles, vectors and fomites.
Right. A vector is a living carrier, a mosquito, a tick. A fomite is any inanimate object that can transmit a pathogen, a doorknob, a stethoscope, an ultrasound probe.
And in a hospital, fomites are a huge driver of infection.
A major driver of healthcare-associated infections, or HCAIs.
To understand how to contain an outbreak, we have to understand the math of spread. And that means R0 and Re.
This is a fundamental public health concept. R0, the basic reproduction number, is the raw theoretical spreadability of a pathogen. It's the average number of people one sick person will infect, but, and this is key, it assumes everyone is susceptible.
It's the maximum potential.
Exactly. And the effective R, is the real-world number. It takes into account all the variables, people changing their behavior, social distancing, and most importantly, herd immunity from vaccination or prior infection.
And our goal is to get Re below 1.
If Re's below 1, the outbreak is shrinking. If it's above 1, it's growing. That's the number we're always trying to push down with public health measures.
And knowing that the R0 for measles is something like 12 to 18 really drives home why you need such incredibly high vaccination rates to control it.
It's just so transmissible.
Okay, so in the hospital, that brings us to infection prevention and control, or IPC. With the risk of an HCAI being around 10%, this is a critical part of everyone's job.
And HCAIs often involve the worst bugs, the multi-resistant ones like MRSA, CPE, VRE, the absolute cornerstone of IPC. The thing you do before and after every single patient contact is hand hygiene.
Okay, but there are two ways to do it: alcohol gel or soap and water. And there's one massive critical exception where you must use soap and water. We have to hammer this home.
The exception is Clostridioides difficile. Alcohol gel is great against bacteria and enveloped viruses because it dissolves their membranes. But C. diff protects itself by forming these incredibly tough spores.
And alcohol doesn't kill spores.
It does not kill spores. So for any patient with known or suspected C. diff, you have to physically wash your hands with soap and water. The mechanical action of rubbing and rinsing is what physically removes the spores from your skin.
Forgetting that one rule is a major way C. diff spreads through a hospital.
A major way.
And for certain bugs, we need to use isolation precautions. There are three main types.
Right. Airborne precautions are for bugs that travel long distances in tiny aerosol particles. This requires a special negative pressure room and N95 masks for staff. Think measles or pulmonary TB.
Okay, what's next?
Droplet precautions. This is for bugs in larger droplets that travel shorter distances. You need a surgical mask and some spacing. Think influenza or meningococcal disease.
And finally, contact.
Contact precautions are for bugs that spread easily on surfaces or through direct contact. You need to wear gloves and a gown. This is for norovirus, MRSA, and of course, C. difficile.
And then we scale up from there. A cluster becomes an outbreak, which can become a pandemic. And that's when we see massive interventions like social distancing and travel restrictions.
All designed to push that effective R number below 1.
The ultimate form of control, though, is immunization, which can be passive or active.
Passive is a short-term fix. You're giving someone pre-made antibodies or immunoglobulins for immediate protection after an exposure. Active immunization or vaccination is about training the body's own immune system to create a long-lasting memory response.
I want to focus on the anchor of conjugate vaccines. Why were they such a breakthrough?
This is a really cool bit of immunology. A lot of nasty bacteria like Haemophilus influenza and Streptococcus pneumonia are covered in a polysaccharide capsule. If you make a vaccine just from that polysaccharide, it only activates B cells. You get some antibodies, but no T cell help and therefore no long-term memory.
So the weak response.
A weak and short-lived response. So what they did was they chemically linked or conjugated that polysaccharide to a carrier protein. That combination can be presented properly to activate T cells, and suddenly you get a powerful, robust, long-lasting memory response. It was a complete game changer.
And the goal of all this is herd immunity.
Which is when enough of the population is immune that the virus or bacteria just can't find enough susceptible people to keep spreading. The chain of transmission is broken, stays below 1. Okay, a final check on IPC strategy. If a patient comes in with suspected pulmonary TB, which type of isolation precaution do you need to implement immediately? And for the 100th time, why must you use soap and water instead of alcohol gel if a patient has C. difficile? Airborne precautions for TB and mechanical hand washing to physically remove those hardy C. diff spores.
Got it. Okay, we've identified the enemy, we've assessed the spread, we know what kills it.
Yeah.
Now it's time to launch the counterattack,
Yeah.
the antimicrobial arsenal. The text lists 5 key components of treatment, but I want to jump straight to number 4 because it's so often overlooked. Source control.
Yes. Source control is often the single most important thing you can do to treat a severe infection, sometimes even more important than the antibiotic choice itself.
What does that mean in practice?
It means removing the focus of the infection, draining an abscess, removing an infected IV line, cutting out dead tissue, taking out an infected prosthetic joint.
Why is that so crucial? Can't the antibiotics just handle it?
No. If you leave behind a huge collection of pus, dead tissue, or a biofilm on a device, the sheer number of bacteria and the terrible local environment low oxygen, low pH. It just overwhelms the antibiotics and the immune system. Drugs can't get into pus effectively.
So you have to physically remove the problem.
You have to lower the bacterial burden so the antibiotics and the host defenses actually have a fighting chance.
Okay, so let's talk about drug selection. We have empiric versus targeted therapy. This is the start smart, then focus strategy.
Exactly. When a patient is really sick, sepsis, meningitis, you don't have time to wait for culture results. You have to start smart. You give empiric therapy, which means a broad-spectrum antibiotic that covers all the most likely and most dangerous pathogens for that particular clinical situation.
You're casting a wide net.
A very wide net. But then, once the lab calls you with the ID and the susceptibilities, you then focus. This is de-escalation. You switch from your broad-spectrum agent to the narrowest spectrum drug that the organism is susceptible to.
Why is that so important?
It reduces toxicity, it's often cheaper, but most importantly, it minimizes the selective pressure that drives the development of antimicrobial resistance.
We also sometimes use combination therapy.
Yeah, for a few reasons. To get synergy against tough infections, like a biofilm on a heart valve, to cover infections with multiple different types of bacteria, or, critically, to prevent resistance from developing in bugs that mutate really fast, like HIV or tuberculosis.
Which brings us to the ultimate threat to our entire arsenal. Antimicrobial resistance, or AMR. It can be intrinsic or acquired.
Intrinsic is just a natural property of the bug. For example, vancomycin can't get through the outer membrane of gram-negative bacteria, so they are all intrinsically resistant. Acquired resistance through mutation or getting a new gene on a plasmid is the thing that's causing a global crisis.
And the text lays out 4 main ways they do this. The first is just destroying the drug.
Enzymatic degradation. The classic example is bacteria that produce relactamase enzymes. These enzymes just chew up the active part of penicillins and cephalosporins, rendering them useless.
What's the second mechanism?
Target modification. The drug can't bind anymore. The most famous example is MRSA. It acquires a gene called MECA, which changes the shape of its penicillin-binding protein, so lactomas can't attach to it.
Okay, and the last 2 involve getting the drug in and out.
Right. Reduced permeability, where they change their outer membrane to stop the drug from even getting inside. And active efflux, where they use these little protein pumps to actively kick the antibiotic right back out as soon as it gets in.
It's a terrifyingly effective set of countermeasures.
It is, and it's why we're facing a potential post-antibiotic era.
Okay, so choosing the right drug is one thing, but using it correctly is another. This is pharmacokinetics and pharmacodynamics, PK/PD.
PK is what the body does to the drug absorption, distribution, getting rid of it. PD is what the drug does to the bug.
And PK failures are common, right? Like a drug not getting into the brain from meningitis.
Absolutely. Or not being absorbed from the gut in a septic patient. But the really critical concept for dosing is the pharmacodynamics, and there are 2 fundamentally different killing strategies.
Okay, let's break these down.
First, you have the concentration-dependent killer. We're calling this one the maximal striker. For these drugs, what matters is getting a really high peak concentration, the Cmax, compared to the MIC. The higher the peak, the faster and better the kill.
And the classic example.
The aminoglycosides, like gentamicin. Because they are maximal strikers, we can give them as a big dose once a day. You get a huge peak that maximizes the killing, and the level drops, which actually minimizes the risk of toxicity to the kidneys and ears.
Okay, so that's strategy 1. What's strategy 2?
Strategy 2 is the time warrior. This is for time-dependent killing. For these drugs, the peak concentration doesn't matter as much. What matters is the amount of time the drug concentration stays above the MIC.
So you want sustained pressure.
Sustained pressure. The goal is to keep the drug level above the MIC for a large chunk of the dosing interval.
And which drugs are time warriors?
The biggest and most important class is the avelaitams, penicillins, cephalosporins, and also drugs like vancomycin. This is why we have to dose them frequently, like every 4 to 6 hours, or even give them as a continuous infusion to make sure that time above the MIC is as long as possible.
And for some of these trail drugs, we have to do therapeutic drug monitoring, or TDM.
Yeah, for drugs with a narrow therapeutic window where the toxic level is really close to the effective level, like vancomycin and the aminoglycosides, we measure the drug levels in the patient's blood to make sure we're in that sweet spot, effective but not toxic.
Okay, a check-in on dosing strategy. If you're treating a severe infection with a time warrior antibiotic, like a lactam, should you prioritize getting a single, massive peak concentration? Or should you prioritize maintaining the drug level above the MIC for a long time? For a time warrior, you absolutely have to prioritize sustained levels, which means frequent dosing or continuous infusion.
That's the key.
All right, let's do a quick run-through of the main classes anchoring the key toxicities. We'll start with the giants, the growth lactams.
Bactericidal, they inhibit cell wall synthesis. The big thing everyone thinks about is allergy, but true, life-threatening IgE-mediated allergy is much rarer than reported allergy. A key thing to remember is that the cross-reactivity between penicillins and carbapenems is actually very low, around 1%.
And the cephalosporins are grouped by generation.
Right. As you go from first to third generation, your gram-negative coverage generally gets better. Third-gen agents like ceftazidime are great for Pseudomonas, but the critical thing to remember is that, with very few exceptions, cephalosporins have no activity against enterococci.
And the broadest spectrum lactams are the carbapenems.
There are big guns reserved for really resistant infections, but the spread of enzymes that destroy carbapenems is a massive global threat.
Okay, moving to the maximal strikers, the aminoglycosides.
Gentamicin, amikacin. Great for serious gram-negative infections. The toxicity you have to anchor is oto and nephrotoxicity. They can damage the ears and the kidneys, which is why we monitor levels so carefully.
What about the quinolones like ciprofloxacin?
They inhibit DNA replication, broad spectrum, good tissue penetration. But the adverse effects are a big deal now. The anchor here is tendinitis and Achilles tendon rupture, especially in older people. Also CNS effects and heart rhythm problems.
And for resistant gram-positives like MRSA, we have the glycopeptides, mainly vancomycin.
Strictly for gram-positives, you give it IV for systemic infections, but orally for C. diff in the gut. The adverse effect to remember is the red man syndrome. It's not a true allergy, it's a histamine release from infusing the drug too fast. You just have to slow the infusion down.
And finally, the complex regimen for tuberculosis.
You have to use 4 drugs because resistance develops so easily. The side effects are major. Isoniazid can cause liver damage and nerve damage, so you have to give vitamin B6 with it. Rifampicin turns all your bodily fluids orange and has a ton of drug interactions. And ethambutol can cause optic neuritis, affecting color vision. These are not easy drugs to take.
Okay, let's quickly touch on the other drug classes. Antifungals. The azoles, like fluconazole, target ergosterol synthesis.
They do. And because they mess with P450 enzymes in the fungus, they also mess with human P450 enzymes, so drug interactions are a huge issue. You have to check every time.
A newer class, the echinocandins, targets the fungal cell wall.
Right. They inhibit 1-naphtha-3 glucan synthesis, which makes them a really good, often safer choice for serious Candida infections.
And the really big gun, amphotericin B.
Ambi is highly effective, but also highly nephrotoxic. It's tough on the kidneys. We often use special lipid formulations to try and reduce that toxicity.
What about antivirals? How does a drug like aciclovir for herpes work so selectively?
It's a prodrug. It's inactive until it gets inside a cell that's infected with the herpes virus. The virus's own enzyme, thymidine kinase, is what activates the drug. Uninfected cells don't have that enzyme, so the drug remains harmless.
That's clever. And for the flu, drugs like oseltamivir.
They're neuraminidase inhibitors. They don't stop the virus from replicating. They stop the newly made virus particles from being released from the cell. They block the exit. But you have to give them within the first 48 hours to have much of an effect.
And briefly on antiparasitics.
For malaria, the key thing to anchor is that some of the drugs, like Primakine, can cause severe hemolysis in people with a genetic condition called G6PD deficiency. You have to check for that before you prescribe it.
Wow. We have covered an immense amount of ground, from defining the players, the cellular pirates, and the prokaryotes with their endotoxin, all the way through diagnostics, the math of spread, and the crucial importance of hand washing.
And we finished with the absolute strategic heart of all this, the need for source control and mastering the pharmacodynamics, knowing exactly why your maximal striker needs a high peak and your time warrior needs sustained time on target.
So to make sure all of this is properly anchored, let's run a final mental drill. Imagine a patient is crashing with sepsis and you think it's coming from a deep abdominal source.
Okay.
Mentally, walk through the whole path. Suspicion, resuscitation, getting your cultures, starting that broad empiric therapy, the start smart, and then getting that rapid lab ID back, and finally doing the single most important clinical step.
Which is getting the surgeons involved for source control and then focus de-escalating your antibiotics based on the culture results. If any part of that sequence feels fuzzy, especially the why behind the drug choice or the need for source control, that's your high-yield topic to go back and review.
We've talked a lot about AMR, and that really provides the final provocative thought for you, the learner.
It really does. The relentless rise of antimicrobial resistance is driven by selection pressure, and that pressure comes from us using antibiotics, especially broad-spectrum ones. So as a future clinician, every single decision you make, your choice of empiric drug, how quickly you de-escalate, your commitment to source control, it isn't just about saving this one patient in front of you.
It's an act of global antimicrobial stewardship.
It is. So the question you'll face every day is this: how will you balance that immediate ethical need to use a big gun to save a life right now with the long-term global imperative to preserve those big guns for the future? That tension is at the very heart of modern infectious diseases.